already mentioned to you that we are having these four speakers in this session on AI and legal aspects, and we'll start with Dr. Saikat Chakrabarti, who is a senior principal scientist and deputy head of the structural biology and bioinformatics of CSIR, Indian Institute of Chemical Biology. Now, as you can see from the PPT, that Dr. Chakrabarti is has got a lot of done a lot of things in this particular area, and what we can say that in this area of bioinformatics and structural biology, he has got a huge contribution already made. So I don't want to spend time on reading all these things in front of you, but I shall request Dr. Chakrabarti to come over and deliver his lecture on role of AI in drug design and development. Over to Saikat.
Good morning, everyone. So at the onset, I would like to thank the organizers, especially Dwaipayan, for inviting me over here in this august presence, as well as in this so-called this heritage building. This is my privilege to be here, and so I'll just go because for the interest of time, I'll just go a little faster. Please brace yourself. So I have to finish it within 15 minutes most likely. So let's get it done. So basically, already this. The platform has been set by Dwaipayan and given this wonderful paper as a brief introduction to what is AI doing and what it can do. This is probably the question is one of the most common question we all are having in different stratas of society. So my thing is be much more subdued. It's basically mostly related to research and its potential to biotech as well as pharma industries. So what I'm going to talk about today is basically hard work of my PhD students, postdocs. So I'm just their mouthpiece. So with this, I'm going to spend a few minutes on basic introduction of AI and ML, and then I'll just share some of the things which has been going on currently at the lab, and we are also trying to see what we can do in next 5 to 10 years in this field. Using AI ML techniques. So my first disclaimer is I'm not a computer scientist. I'm a biologist. I turned into this field by mere accident, and then I fall in love with this field and slowly moved to this last 10 years to machine learning and deep learning and artificial intelligence. So these are terms you'll be hearing a lot, and probably you have already heard a lot, and you have already made a different, probably 100 types of definitions of what is AI. Okay. So everybody has its own definition, but believe me, it is simply just a tool. It can do something, but it may not change the whole world. It may not change our perspective. It's just a tool. All it can do, it can probably try and simulate our mind, and that's why it is called artificial intelligence. And probably that's why people like me who lack natural intelligence, they are actually getting associated with this. So it can never replace natural intelligence. All it can do, it can try and simulate, maybe not as accurate as the natural intelligence can do, but for example, it is broadly defined as a technology that can learn and produce intelligent behavior. In this case, let's say human behavior. There are subhuman intelligence also, but in this case, it can try to simulate human intelligent behavior. For example, if it can do looking at a chest X-ray and identify the disease, which is for us, when a specialized human beings, they do it, and it's a years of training and experience. So if challenge is if AI can do it successfully. Similarly, from patients, if derived different biomics data, which we are talking about, one of the topics is genome and genomics, and it's because the omics does not stop at genomics. There are other omics data. For example, it's not the genes only. There are a lot of things, a lot of complications there, a lot of molecules there. For example, there are RNAs, there are proteins, there are metabolites. All these they are playing a role, a concerted role in keeping us healthy. And if something goes wrong, and then that could actually turn us into unhealthy or disease. So the challenge is, how do we try to understand these complex scenarios and see whether there is a pattern which is differentiating between healthy versus non-healthy or diseased. Okay. So AI may be able to try and do that, because it is not possible humanly to handle these large amount of data with large different complex interrelationship between the data and between the biomolecular type. So we will talk something about that, or even another part which is again directly related to today's topic is drug discovery. Okay, drug discovery is inherently related to biomolecular interactions. For example, the drugs, they do interact with biomolecules. That's why they are called drugs. Similarly, our workhorse are proteins. Proteins work by interacting with each other. Okay, so they exhibit their functions by interacting with each other. But the knowledge about these interactions are best known by experiments, by looking at their structures, the three-dimensional shape and interaction type. But those experiments are humongously costly as well as a lot of time consuming. Because today, in the need of the hour is we need to know the information much faster in a much cheaper way, so that we can, remedy can be given faster way. So there again, AI kind of tools or computational genomics can play a role and try and correct or give an accurate structural overview of biomolecular interaction, which could then further be taken into actual production of the drug and testing of different phases of testing of the drugs. So it is playing a crucial role. So that's what AI is. But what it is it is unit-wise. If you think of it, because the word intelligent is critical, because it is coming from our nervous system, and the nervous system unit is neuron. And you all know that neuron needs input or inputs, and then it delivers an output. Okay, so in this case also, this is an representation of a neuron, where the AI processor takes multiple inputs or single input and provides an output. The output could be a decision. So it is basically a process which is a mathematical function, but the inherent behavior is simulating the neuronal behavior. Okay, so there are different steps of AI, which I'm not going to details, but most important thing you have to say, because it learns from existing informations, existing data. So data collection and data processing is very critical for machine learning, as well as because machine learning is inherently related to artificial intelligence. And so the processes, for example, preparing the data, choosing an appropriate model is critical, and then there is a rigorous training process going on. Once it is optimized in the training, then it can be used for any prediction. Now, these predictions are usually biphasic. It says yes or no, or if type 1 type or 2 type. Now, because it has improved a lot, these days it can do multiple types of predictions. But inherently, it is a two-step process. First step, it learns, and better it learns, better it can produce. So that's what the lower panel tells. It takes the different data, learns using different phases. What I'm telling it's not like it does not learn by itself. We human beings try to learn themselves. Okay, so you have to keep in mind, it's not science fiction. It's human being. We have to spend years. For example, something I'm going to talk about, our models, which we have to spend months and years to train. And if it does not train well, we have to go back again to the blackboard and then redo things, hyper-tune the parameters and all these things. So it is still human beings which are regulating AI. Okay, not AI is doing the other way. So today's topic is that drug discovery and AI. Now, it requires, as I said, the data, the information which is created by humans, stored in not so systematic manner. So that's why we need machines to collect different information, kept in different places, and then put them together to make sense out of it. Okay. So for example, we have sequencing data, which again coming out of the genome sequencing. And it's surprising if you see, because I just thought of, it's almost a quarter of a century since the genome sequence, human genome sequence has been done. And unfortunately, or fortunately for AI machines or people like me, we still don't understand probably 10%, or still understand only 10% of it. So there are a lot of things to do, a lot of things there, and it has been proven that even the best of the human brain would not find out the things which is hidden inside it. So maybe a bigger machine, a better trained machine with a better efficiency can find out something which we could not do. And that's what this next 25 years is probably going to be revolutionized, because this is where they can find out. Because I still remember during my PhD days, there were 99% of the genome were called junk DNA. These days, that's almost a curse. There's nothing junk. Only thing, we did not know what is good or what is bad. So this kind of systems may unravel the unknown things, and that is what is going to is happening these days. So this sequencing information, this experimental data kept in text, kept in someone's textbook, someone's spreadsheets, machines or these programs they are capable of collecting them and putting them together. Similarly, molecular structure. This is a huge chemical structure field. Not everybody can understand. Even for people, biologists like me who turned computation scientists, they don't know. But machines can, because it has the inherent capability of, if I define what is chemical structure, what is chemical space, it can. So that's what it is doing. And it is learning. Learning in the way I give the features to them, and it learns and identifies the subset of the features which are probably most important for delineating two things. Only as one thing I just want to tell you, till now, at least the predictive AI, it can delineate, it can differentiate between two classes or more than two classes. What again, Dhrubada mentioned, there is something, creating something which was never there, which is part of generative AI, which is still in its juvenile state, but I think with more regulation, generative AI is going to be prevailed more than predictive AI. But for drug discovery, right now still it is predictive AI. So now what it predicts? It predicts, probably it can predict the disease diagnosis, because first of all we need to identify what is disease, which individuals are disease, and then once the disease is diagnosed, then what are the probable targets of the disease? Because in drug discovery field, we always need to know the target, because that's what we learned in last couple of centuries, that if we develop drug without knowing the target, we are going to land in trouble. And that's what we are going to having these AMRs. We are going to having these normal cancer drug resistance, because we did not know the target that well. We did not know how it is interacting with the target. So that precise knowledge is actually important to know what target and then do the screening. So these three parts, applications are critical. Then there are so many more I just thought of, because I am associated with some of the things, which is basically the first three: target identification, which is critical, then the target processing, because if I do not know how my molecules are interacting with the target, then it is bound to be failure. Because you know why we have so less drugs, because most of the drugs, they fail in phase 2, they don't even go to phase 3. Okay, so that's because the initial steps at phase 1 is dependent on in silico screening, and the in silico screenings are faster, but it comes with a price. They're error prone, and which goes to in vitro screening, it fails, and even somehow they passes to the in vitro screening, they fails in preclinical steps, because we did not know how exactly it was binding. So this is the challenge of AI and computational screening, to know more in-depth knowledge in binding. Anyway, so from the introduction part, I'm just quickly next 5 minutes going to say what in our lab we are trying to do. First step is to look at identify disease markers. So there are two kinds of disease markers. One is diagnosis purpose, whether I am diseased or not. If I am diseased, which stage of my disease is this? So the first part is if I am diseased or not is called diagnostic marker. All of us, we go through, for example, we measure our sugars and something. This is a diagnosis marker. But then how bad I am in stage of disease, that is called prognosis markers. So using patient-derived different kinds of omics data, different, as I said, that's how we call multi-omics. Probably you've heard this word a little bit now, pan-omics or multi-omics. Omics means, again, larger number of gene sequencing information or RNA expression information or proteins which we have currently working, all the proteins which are working there, their abundance, as well as the metabolites, the small molecules. Our body has small molecules which actually plays an important role in different interaction types, our normal functioning of all different things. If we put them together, that's called systems biology. If we put them together and try and find out whether a certain population, which are cancer patients, for example, whether they have a certain kind of signature markers compared to healthy, which is different. So that's the challenge of identifying these different layers of information, putting them together and identify and cross-correlated signatures markers are there or not. Now this is the biggest challenge is first of all collecting the data, which is these days because experiments have also improved a lot. We can actually identify within a day all the 20-25,000 of genes, their mutations, their expression patterns, the proteins and everything. It can be done within a day or two. Now putting them together and making sense out of them is the most critical challenge because it is huge in size. Now as well as there are complications in interrelationship. It's not like A goes to B, B goes to C. There are interrelationships we don't know. So now how they are regulating with each other, how regulating they are being regulated by each other, it is critical. And that's what we are trying to do in our lab, where we've collected data, these omics, different types of omics data from larger amount of patients, more than 100 patients. And if we look at it, the rows are patient numbers and the columns are different omics data types. If we put them together, they can go into lakhs, 4 lakhs, 5 lakhs of data type. Now this is representing cancer patient as well as non-cancer, or within a cancer patient something which is early cancer stage versus late cancer stage, because these are all very prominent clinical questions as well as social questions, whether the patients are at the early stage or a late stage, as well as whether the patients are having any markers which could be easily used in next as a diagnostic test or something. But then if they are in lakhs, there is no point. We need to reduce it to a smaller number. So our first target was to bring them to around 10 such things, which these markers which are successfully differentiating between either healthy versus cancer patient or early stage versus late stage. And thankfully after, again, this I was talking about, this took us almost more than a year just to handle the data and then learn from the data and then optimize the models. And for example, we working with these different multiple cancers types. For example, 5 cancers data I'm talking about, and I just wanted to show the number of data points. 4 lakh for each patient, and there are 100 patients. So you can multiply, see the matrix size. Even the very, very large and super computer, high performance computer also fails. It takes months to learn that. And then we realize, no, it did not learn well. So to go back again and redo, change the parameters and do that. So, but then at the end, we could say that we have successfully trained these models, which is now capable of, if I give a naive, which is not been seen by this model, a data type taken from a patient or not, it can successfully tell me whether it is an early stage cancer or a non-stage or late stage cancer. So I'm just, for example, showing a data of colon cancer, where if I had taken all the parameters, I don't know how to point it. Anyway, if you look at all parameters, the performance matrix is not good. That means I've taken all the 4 lakhs parameter and trying to differentiate between early stage, for example, stage 1 versus stage 2 or stage 1 versus stage 3 of colon cancer, and it is going around 50% success rate. But if I go to 20 or 10, the success rate goes, accuracy rate goes around 80 to 90%, which is quite acceptable at the clinical stage. So in a way, we have not only reduced the numbers of the features, but also improved the accuracy of delineating or differentiating. So now these markers could easily be used as I said, diagnostic marker, prognostic markers, or therapeutic markers. That's what we are trying to do, but obviously we cannot stop here. We have to testify in lab, in our setup, whether what we are telling about, whether they are, what is the abundance levels of patient and whether they are statistically significant or not, and then of course then go to clinics and approach industry to try and set up whether they could be used in real life or not. But anyway, moving next maybe 5 minutes, the second part I was talking about, the molecular interaction patterns, both at protein-protein interaction as well as protein-small molecule interaction, which is directly related to drug and target interaction. These are, as I said again before, that experimentally they have solved, but even experiments can have error, as well as they are mostly these days are solved by computational tools and techniques. Molecular docking, I don't know whether you've heard something, molecular docking. This is a computer derived how the 2 proteins can interact, or how a small molecule can interact with another protein, but they are error prone. That needs to be validated whether they are good or they are just throwing them away. So what in our lab we are trying to do, both for protein-protein interaction as well as for protein-small molecule interaction, we implemented machine learning kind of techniques. How? We believe the experimentally solved structures are actually representations of the correct molecule, because it is quite rigorous, and there is no, one can argue that they may not be the best, but they are, represent the actual true complexes. What we try to do, we try to simulate that using molecular docking techniques, and we created some, for example, we took an already solved structure and then we took them apart and we put them into a molecular docking, and then they were reattached together. Now we screened all those hundreds or thousands of solutions which are very similar to the originally solved structure. So that we call them as native-like structures. So they are native-like structures, which are non-native-like structures. So therefore we created 2 different classes, which are true complexes and false complexes. And then we calculated their features, the interface features, because interface is where the 2 molecules can interact with each other, and the features which is representing the true-like or native-like, they will have their distinguishing properties respect to the non or false-like structure. And that's what we did. We learned, trained the model based on these 2 features, and thankfully, and then tested again. And thankfully, if you see the table, if you can see the numbers, I don't know, we have reached more than 90% AUC values, which is very, very good. If I've been, so encouraged by that, we have actually created a web server. So anyone having a protein-protein complex, either coming out of an experiment or crystallography or NMR or even a molecular docking, can actually put them here. And within few minutes, we can at least tell with a probability that whether it is likely to be a native-like structure or not. If not, maybe they can recheck and redo it. So and thankfully, this has become quite popular, and this is one of the most downloaded programs and access paper of it. So hopefully, we can improve it better in the next few years. Anyway, so last part, protein small molecule docking. Same problem exists over here, because molecular docking, in vitro, in silico screening is a critical part of drug discovery regime. and there are errors inherent, errors of docking programs. So how do we correct it? We went, in one approach, we tried to create newer docking programs, solved, use multiple docking programs, which we are working, but at the same time also we are trying to do, even using one docking program, can we learn from the known complexes and try and find out features or characteristics which are representing the actual known small molecule and drug form. So similar to the protein modeling we have done, we have created different scenarios of how already known drug or drug-like molecule is binding to a protein molecule. Okay, and we created native-like structure whereby redocking them, and also created which are not like native-like. For example, some same small molecule is binding to the same pocket but different way. That's we call false complex. But the same molecule can bind to a different pocket altogether. Okay, or even the small molecules can slightly alter, can get altered. All these things scenario, or even the receptor itself, because in disease, if you know, most of the time, by virtue of genomic understanding, we realize that in diseases like cancer or any other genetic diseases where genes get mutated, and because of the mutation, the drugs which usually used to work with the wild type, now they don't work. Now, can the drugs which were there, or can we design newer drugs which could actually identify these mutated proteins? So that's what we've also created scenario where the protein is altered, but the drug, whether they are binding it or not. So all these different scenarios put in together, we have created a package where this generation of hundreds of models are there, all using all these scenarios. And then ultimate goal is, if I have a complex out of again molecular docking program or a crystallographic results or even other things, can I test it? Can I tell with a confidence value that this looks like, it's very similar to a known complex, so maybe you can go ahead or not, otherwise no, go back and redo it again. So again, thankfully, after years of struggle, we have got into a system where different benchmark told that we are getting more than 90%. Here, 90% is very critical, and that's why I see in some of the values we still around higher 80s, but we need to create it and improve it. So we are improving it by putting more complexes together and creating more features where small molecules. So right now this is almost 5,000 features. So if I can improve more features by defining how a small molecule in interacting with a protein, and this, believe me, this 5,000 is a lot. We may not, but if we define more accurate, sensitive features, the accuracy can go higher. But anyway, this was good enough for us to file the initial patent screen. So right now this is basically a machine. So we have put this package into a machine. So that because in India you cannot patent a software. In US or in abroad you can do, but that's what we had to do. We had put it into a computer machine, and that device is now being patented. So once it is patented, then you can testify in a much larger scale. So that's it. So, but just take home message for you, AI is very, very advantageous. Believe me, I've been in this field for last 5-6 years, most rigorously, but because of its speed, because of its chemical space exploration, data integration, and of course the cost effective. It is much more cost effective than even for the traditional drug screening method. Okay. However, it is limited. There are challenges there. First of all, the data quality issue. One needs to be smart enough to take which data to take, which data not to take. Okay. And it is kind of a black box. I don't know how many of you know, because most of the time we don't know why it is giving me so good answers or good accuracy. That's why all ML or AI models are inherently, there is a black box part. I'm not going to detail, but if you want to talk, I can talk about this black box, which is interesting. And of course, there are regulatory concerns, especially in case of biomedical research. Rampant use of AI is a big threat. Is a big threat. So we have to be we only can actually regulate ourselves. It's like the same threat we used to have in our school days, science, whether it's a cars or it's a so here also you have to use AI as a tool, not like answers to our all problems. So regulatory concerns are there. Future direction, explainable AI, which is called XAI, because that kind of attends that black box problem. We need to know, as a human being, as scientists, we need to know why it is doing so well, why it is differentiating. So we need to identify the features which are differentiating between two classes or multiple classes. So that's what explainable AI is going to come. It is directly connected to the computational power. So quantum computing is going to be revolutionized this field, because believe me, this is huge computation. It takes months on a very, very highly powerful GPU system. But quantum computing is the key to actually help this to actually solve real life problem in real life scenario. But so far, there is one thing which is we taken for money. For example, Bangla, there is one thing called Dudher Shad Ghole Mithai. So federated learning has come, which is nothing but using multiple computers spread across all over the globe and utilize them together. It's called Internet of Things. I don't know whether you heard the call IoT. So using those computation resources, even for example, your mobile or can be, if you give concern, I mean, it will come in next 5 to 10 years, or even right now they are doing it without our knowledge. It has been their hardware has been utilized so that combined computational power is being utilized for these things. Okay. For example, if you do a Google search today, and then next, after 1 hour, if you do a Facebook, you'll see that same thing kind of search is coming to the. How do you know it is coming? Because then means there is a connection to your things. So that has to be done. So that's the federated learning is going on, and of course AI guided lab experiments. So these are the, I mean, as needless to say, this is collaboration is very, very critical for both academic, industry, and clinical, because the field we are talking about solving, diagnosis disease and solving disease conditions. So, and there are multiple companies. I am just listed few of them which are in this field, and of course, this audience, they know much better than how much the computation and AI is coming into this field. So, thank you, and I'm going to take your question if you have any.
Thank you, Dr. Chakrabarti. And what I was thinking that because all the talks are of the similar in nature, so maybe at the end of this session, we are going to have the interaction. So keep the questions in your notes, and we can go for these questions. And so, Dr. Chakrabarti, thank you. And we'll move on to our next speaker, Dr. Dwaipayan Bharadwaj. And actually, you can Dwaipayan Bharadwaj, who is actually the reason why we all know, because of his team actually conducted the India's first genome-wide association study on type 2 diabetes. And he actually founded the Indian Diabetes Consortium, the largest collaborative model for this diabetes research. And he was last 10 years in Jawaharlal Nehru University. And just this year, he joined Sister Nivedita University as senior professor also. So he's going to tell us about the AI in diagnosis in genomic research. Over to
Hello. Hello, everybody. Thank you, this Bengal Chamber of Commerce, Professor Dhruva-Jyoti Chattopadhyay, to invite me, and giving me opportunity to learn AI. So it's obvious that these things I'm also learning when I'm preparing these presentations and others. And when I'm preparing that, I think that something in a very loose way that we can say that we know what is AI. To me, it's a rote learning. No? So that from very early childhood, see that in my grandma's mouth I was learning Duradas Chakro Nivashatani Tamalotali Banraji Nila Abhati Bela Labaram Budashe Bodhara Nibaddebo Kalanko Rekha. So our whole life is learning rote memory. How far we can learn this rote memory, and then we analyze, and specifically Indian system of education is like chalo rote memory and then learn. So it's a pattern recognition. So imagine, so this kind of rote memory, like say 20 or 20,000 human brain together is analyzing and coming to you. Because this rote memory is a kind of genetic, that whatever my previous speaker is saying, so I am taking a clue from that. So if you see, it goes to the family also, the rote memory. I heard that my grandpa, he memorized that entire Milton's Paradise Lost. So he could say that this page has this thing. So what AI is doing, actually we were doing AI, because as professor said that we were doing with the genome-wide association studies, there's also 600,000 SNP, single nucleotide polymorphism, and 20,000 people, so that many data point, and then showing that which SNP or which mutation is associated with the disease. It's very easy when it is one gene, one disease, 5-6 gene, then you know, it's a pattern recognition. Imagine that many of our neuronal diseases are triplet repeats. So TCA, you know that 40 up to 40 repeats is normal. So when you changes that 50 onwards, then you are finding schizophrenia or that kind of neuronal diseases. So now you imagine this pattern is not continuous. This pattern after 1 lakh point you are finding. So at that point, human brain cannot analyze that, because we cannot see the full things. So this is a simplified variation, that pattern within that millions and millions data. If we use this trained human brain itself, like those, I say, this artificial intelligence, then pattern it can discover very quickly. In a nutshell, that is that AI which I learned when we are doing that. The story of this DNA diagnostics and AI starts with this 2000, when those Science and Nature came with that human genome, because you need a data set. As Saikat shows, that there will be a data set which is test data set, and there will be another data set that is trained data set. So this data set in our cases, this human genome, and human genome we are learning every day. As he also mentioned that junk DNA. What is junk DNA? Junk DNA, people were not working at the time because Sir Francis Crick told them as a junk DNA. Francis Crick, Nobel laureate, telling it's a junk DNA. So almost a decade, nobody ventured in that area to analyze pattern or anything. At least more than a decade after that, when Francis Crick told that, people realizes that's a golden mine, that you can get some pattern and some things like that. And specifically, we know that intron and other, that huge portion, that human genome 1% is the coding. So more of them we do not know, but I can say that's an evolutionary deposition for the variation, because nature never select individual, never selects the whole species under his or her care, because nature believes in species extension, not individual extension. So it's we know from 2000 onwards these things is going on. And if I say in a cartoon wise that this huge data set where they are keeping, that I say that individual human can analyze a short genome or small area. Then comes this, I'm showing the slow stage of this going to this artificial intelligence. Then we started creating supercomputer. So supercomputers at those days are actually in our desktop. So whatever at that time was supercomputer, now we are using on our desktop as that those we called workstations. So you imagine that one earlier only some, what I will say, not even a 1000 bytes data, there was those memory sticks. But in now one memory sticks, you are having terabytes of the data. So analysis part is totally different. So during this human genome sequence, there were 6 countries, 20 centers, more than 10,000 researchers. They discovered that thing, and still we are doing this, and ultimately 2000 this paper came. It came modified in 2003. Then starts the big data analysis, because before that nobody has that big data analysis, that whatever we are doing now, AI is being applied in stock market, in solar system, everywhere. But it starts its journey, the big data journey, from the human genome sequencing. These biologists, initial human genome sequences was not done by biologists. These are all physicists. They started in the Utah mountain to do these human genome sequences. So they offered us first time this big data. And from 2000 onwards, initiation of this AI started unknowingly, that human genome part. So then we are starting that rapid decline in the sequencing cost, because sequencing earlier was a humongous costly. The moment is now within $1,000, one human genome can sequence. I think it's lesser than that. Now you have nothing, even in government of India, it has a project of those 10,000 genome or something like that. So initially worldwide it started like 10,000 genome, now already they finished more than 100,000 genome. Now you have those cost per raw megabase of DNA sequence coming down, and you have the big data. And now people are starting meaningful data of that. So just it coming every day. It coming every day means, because I deposited this stock at least 1 week back. Within that week, I find this thing. I thought that it will be interesting to you. This is a picture of 1 cubic millimeter of brain tissue. You know, 1 cubic millimeter, that in my brain, that 1 cubic millimeter brain tissue is here. And this picture of this human, this 1 millimeter, 14,000 4K movies. All of you are acquainted with 4K movies? That movies with the highest pixel size these days, in the highest quality HD, we are seeing in the OTT platform. So that 4K movies, 14,000 4K movies are in this 1 millimeter. Earlier we know that neuronal and neuronal connections are like those dendron and other things. The modern technology, this science when it is developing, as the engineering and other tools. One science's new discovery is other science's tool. So now we are seeing that if you see the picture, this is a 1 cubic millimeter of human tissue, and which has this 14,000 K of movie. So human brain cannot do that. That's why we made a tool that this is human being. We are making this demon or this is angel for our analysis. And eventually where it will go, we cannot predict, because whenever we see the science fictions and other things, then we are finding that artificial intelligence is such, because it's intelligent. So automatically within the system, it develops, it finds its own path, because those life always is dynamic. So we are trying, the scientists, researchers, unknowingly and knowingly making this artificial intelligence, thinking it has a mind of its own. I will not say it is a life. So scientists captured this detailed 3D, as I said, this map from the brain tissue, which is so humongously important, and we can do those analytics. In definitive scale, if I see these all are those ATCG, and then from there that we need to determine the patterns. Can human brain determine those 3 patterns? Answer is no. There comes our AI, because the next generation of diagnostics will obviously go to this AI powered diagnostics, because initially when cytogenetics came, the all diagnostics was chromosome based and under compound microscope, and then came the human genome sequencing, antibody interactions, ELISA. Slowly if you see that our power of this diagnostic is going up, up, up. So the moment we will find this pattern recognition in human system or in life, it will give a powerful tool to predict future of the disease. And then instead of now what we are doing, we have a fever, we have something, we go to the diagnostic centers and then they give you the diagnosis and then physician will start the regime. But now if I can predict that this situation, this part of chromosome is vulnerable for some bacteria or virus to interact from very beginning, then the precautionary role of the diagnostic medicine will start. So world will slowly focus onto that. New reality is that data is growing, so it's storing. It's stored somewhere because until and unless we get the powerful tool, and that one kind of powerful tool probably we get. So now AI will be automated and it will be intelligent interpretation. If we see not only even the biology, if you see that driverless cars. So what is driverless cars? How that driverless car accumulate all of those informations of a crowded road. So it's already there. We are finding, we are taking from those technological breakthrough from that area and started applying in our biology. So these slides, if you see this, you will find some of those slides I was finding very interesting, because I was also looking some of those slides from BioRender and other things like that, and unknowingly or knowingly, AI probably driven that some of the slides I found also in the same way in the net whenever you search with the AI. Because if you find these days that in your mobile, if you search something, so 5-6 days after, if you want to do something and if you put ABCD there, it will automatically go and giving you the clue that these things you are searching. So already your mobiles and everything are AI powered, knowingly or unknowingly. So this is going that with this massive data and how to give a meaningful patterns. Those patterns are hidden, those patterns are not in front of you in a very straightforward. That is the artificial intelligence in genomics. And what is artificial intelligence? Is nothing but those analytic power within that huge ocean of data. That's what is doing. That you have a set where you are helping your computer to understand this pattern. Automatically you are generating another pattern that this machine learning can be done because already computer got the cue, and next part of your analysis, it will be aided like that. So how it will help you? Say when we are doing this drug discovery and other, first to tell you, telltale sign is that drug discovery is not university's job or a particular individual's job. It's a group of people are doing that. So those group of people with different expertise will be more beautifully integrated through AI. AI will cut cost of your animal experimentation, because animal experimentation, we are now statistically thinking that 6 control and 6 animals in the experimental will be the best. But AI can say with all of those animals characteristics, no, it can be done with the 14 because these are the data available in these 2 animals. It's very simplified thing, but in that way, AI will cut cost also during your experimentation, during your animal uses. In that way also, it will be wonderful. And see that AI-based things we already started using. See, one of the things is like that, for the last 3-4 years, probably we are using ChatGPT. That is one of the massively used AI-based this thing, and it's very tricky when we are using ChatGPT also, because ChatGPT has its own mind. Different kind of robotics are coming, specifically in the surgery rooms. These are AI-driven. But AI-driven means backside there are human brain, because when it is making that human brain started and purifying it to that accuracy, and then we are using. So if I go to this machine learning, then you will find that there are different stages. One is that artificial intelligence, and then the machine learning, and then comes that part which, as a researcher, which the particular researchers of the field, they also don't have the 100% clue that how this deep learning occurring. Because in our case, in human brain, we say that you first learn how the learning process comes, you read that information, and then when you take the information, you apply in another field or in some way in your life, then it's called knowledge. So this is the stage of information to knowledge. And that knowledge, when you are using more time with age, it becomes wisdom. So wisdom is not quantifiable, but this jump from information to knowledge is very easy and trainable. So I think that is the thing artificial intelligence telling, that machine learning, that artificial intelligence to machine learning, that is that information and jump to that your knowledge. But deep learning is like those wisdom, that whenever this machine is doing that in the artificial intelligence machine, and that within the system it tinkers such, you are finding this deep learning. What reflection we get in some of those science fiction, and really I don't understand also. So if you find that a features are feature selection, and if it is machine learning, that is manual and expert driven, and expert is that human being, and deep learning is automatic and learned by the network. But those network individual components are developed by the human brain. And data requirement, in case of machine learning, we have two sets, one small to medium. But the moment we are coming using artificial intelligence and deep learning, then we need the deep data set to understand all of those big patterns. And computer need, it's a PC will do your machine learning. But these days, supercomputers in the background, you need this modern deep learning or this artificial network. And transparency, you understand the algorithm in case of machine learning, which is easier to interpret. But according to the deep learning thing, it's often now, it's a black box. We still do not know what kind of patterns are coming until and unless we understand those pattern background, it's difficult to decipher them. So reasoning is but beckoning. How the human brain inspires this artificial intelligence? If you see that recently it comes the biomimetics. The before brain, scientists started that, you know, the best of the adhesive these days are best of the adhesives. Okay, okay, I will I will go a little faster. So best of the adhesive, if we see these days, these are biomimetic. People see that grasshoppers, they jump on the tree and attach themselves. It's with a very decisive attaching. So what scientists did, they took those things and they did this cytological staining and different microscopy and find out the molecular position of those particular addition of those things. And then chemists in the lab used the same molecular biological pattern and came out with the best of the adhesive these days. So same thing happening here, that human being inspires the artificial intelligence, because my earlier speaker also showed that how the brain's network of neurons are, in this case, computational systems we are doing. We are learning mechanisms, we have the cognitive functions, and third is the memory and recall. In the same way, introducing the artificial genetics in genomics, artificial intelligence in genomics, we have the big data set as the human genome sequence and the entire of those sequencing. And how does it work? Because all of those genomic sequence in front of us, so we have a humongous data set for those training mode, and that training mode is generating the AI model, and that can be tested. Then initially what we do in our laboratory, we collect blood, we test, we tag with the genetics and the medical test. All of those goes towards the building the model. And now the same thing we are doing, giving the same diagnostic depending upon the what kind of medical test we did, and we give the treatment option and prognostics. So this thing now, if I show you this chart, you will see that application will be in the same way, but everything will be deposited in that AI network, and it will show the particular patient's disease, because background all of those genetic information is already there, And in the same way, they will do the rare disease diagnostics. And moreover, with the huge data set, it can find out the pattern, and those patterns will be the biomarker discovery, which these days we are doing, hunting the different pedigrees or going for family to family and collecting blood. The same system, when it is already deposited millions of people's human genome sequence, the biomarker discovery will be easier. How do AI improve diagnosis? Obviously, because AI will automatically remove those which are removable and only keep those important ones. And it will give you that this particular person will not work with this drug because it's already shown in his diagnostic feature. So it will cut the cost. So from complex disease research to clinical complications, it's very important because complex disease, they come not only as a genetic, it comes with the epigenetic and other environmental ventures. That's why they are complex. And these genes, and with the anthropometric parameters, using this AI will be much easier. I will not go into our data as per se, because cases and controls, because for this time short. So what we will do for those thousands of analysis, we can have a polygenic risk score, and that will give you the high risk and low risk. And then depending upon that, you already are finding new companies are coming. They are showing the rare disease diagnostics, the phase 2 gene, they are a 90% accuracy, because diagnostic case, if it is not more than 99% accuracy, then people will not buy. Say, I have a life-threatening disease. If it is not 99.9%, who wants to know that I have a 70% or 80% risk? So that jump from 90 to 90% will be helped by these AI-driven softwares. And in that way, sometimes maybe precision medicine comes, because precision medicine in a country like us is a dangerous thing also, because precision medicine will be highly costly. So I will skip this, but I will not skip this one. So what is that use with the caution? It's very important that recent paper shows this. Listen carefully, because this is only 3 years ChatGPT people are using. And there are people who are using ChatGPT continuously. This paper came, I believe in Nature, that 3 weeks back. They showed already the people who are using day and night ChatGPT and the people who are not using ChatGPT in their profession. Actually, this is the position of their brain. When they did the MRI of the brain, they said the people who are using left, right, central ChatGPT, their neuronal sparking is much less than people who are still thinking and doing their own work. So what I will say like Buddha, it should be a Madhya Pantha right now, because this is again we are showing in front of us evolution, adaptation and evolution, because we may going towards another evolution of this when we are using rampantly something which we do not understand clearly. So this is you're fresh from the oven, those things. And these are the people who work. And I put Praveen Gupta all the time because he's MD of a company, because he is the Illumina CEO, Illumina company CEO in India. So all the time, if I require the experimental help, experimental reagents and those things from him, I usually get. And all of this physician, without physician, I couldn't have data, but I didn't show you the data. And I'm endowed earlier when I was doing experimental research with large funds. And this was that all the time the students came from my laboratory. So initially there were 3 person I want to mention. Those, I don't know how to show them, that you see that there are 2 boys and 1 girl, almost in the middle, rightish. Anubha Mahajan, Amitabh Sharma and Srinivas. Those are my first 3 students, and actually at a time they were the faculty of Oxford University, Harvard University and Cambridge University. I think that's the greatest achievement at the time I have. And if we have questions, what Sir says is the last I will do.
Thank you, Dwaipayan. I think that we have certain ideas which are coming out of this particular presentation, where we can discuss a little bit afterwards. But we'll move on to Dr. Sayak Ganguli. Dr. Sayak Ganguli, who is right now the assistant professor in St. Xavier's College, and is going to present before us regarding the genomic data analysis powered by AI, opportunities and challenges. Over to Sayak. Sayak's expertise is in the area of plant.
My basic expertise lies in the plant, but I have been doing bioinformatics for the past 10 to 12 years. So good afternoon, everyone, and thank you for the opportunity to interact with this august audience. Thank you, DJ Sisa, for inviting me. And so far we have listened to two invigorating talks where we have been introduced to the various aspects of AI. What I intend to do is to give you a reality check, because the use of AI is not universal, and the use of AI is often infrastructure driven. So one of the key things that we need to understand is, if we are looking at the data that we are looking to analyze, we are looking to analyze 40 exabytes of data, and that is equivalent to more than 150 billion textbooks. That is the future of data that will be generated through different kinds of genomics experiments. And all the faculty members that are present here in this audience on the dais will agree that we do not have the necessary infrastructure available to us at our individual institutes, and hence there is an essential requirement for setting up hubs where we can actually access, students, faculties, researchers can actually go and access and perform experiments which cannot be done in the individual institutes or labs. So that is one of the key aspects that I intend to start my talk with. So this is the kind of data that we are looking at. Next, when we are talking about data, we are talking about the variety of data and the veracity of data. So the variety of data requires different data models, because different experiments will generate different kinds of formats which needs to be analyzed. So when we are looking at different formats which needs to be analyzed, they need to be set up in a specific relational database model. So when we are looking at relational database models, then there has to be specific access points through which we will be able to access them. So when we are looking at, say, a Google search, we are essentially doing a text-based search. Similarly, I believe majority of us has searched for some terms using NCBI database. So again, NCBI also allows us to use a text-based search. But what is the fundamental difference? In NCBI, you can use some Boolean operations to screen your search. How is that functional? Because NCBI has a background ER diagram and a ER model which is built upon a very robust relational database schema. But look around us in the Indian context. Do we currently have something which is merely at least 50% efficient as NCBI? We currently do not have anything such as that, because we have not been able to develop a model which will enable us to share the data that is being generated. Again, whenever we have to get an accession from, we have to submit all our sequences, the whole genome sequences that you are generating, the transcriptome data that you are generating. We are going and submitting to UniProt. You are going to submit to NCBI for generating the accession, which is essential for publishing our papers. So technically, we do not have the infrastructure, the computational infrastructure or the analysis infrastructure which is accessible globally. So that is something which is fundamental for future of AI-based research, where people will have access to infrastructure. And that is something which is extremely lacking. So if you look at this particular slide here, this gives you an idea
in terms of understanding what are the various different types of data that we are talking about. And every data point that we are talking about here or is mentioned in the slide has different kinds of analysis platforms that needs to be looked into. And hence the requirement for data models. So that is something which is of paramount importance. We have been talking about the human genome. But what about the other genomes? Specifically, DGCSR mentioned in his opening talk about the fundamentals of increase in antimicrobial resistance. So what about the other partner to that? What about the microbes? There is a deluge of microbial genomes that is available in online resources. And yet, we are not able to understand which particular biomolecules are we going to target, because as soon as we develop a drug against a particular biomolecule, the gene for gene hypothesis takes us to the next level where the microbes evolve. And automatically, the drug that you have developed becomes useless in a span of 3 to 4 years. So there is the necessity for exploring the microbial genome space as well. And not only are we looking at basic genome analysis, we have to put in the evolutionary perspective there, where we are looking at the pangenomes as well. Because when you are looking at the pangenomes, you stand the chance of understanding how a particular microbial system has evolved across time. And when you have the opportunity to understand that evolution across time, you probably have the chance of identifying a large number of extra targets which can be used in your drug discovery process. So one of the key aspects that needs to be kept in mind is not only, yes, human genome analysis is something which is extremely important, but we have to look at genome analysis of other partners as well. So that is something which has to be looked into. So we all are aware of the two DeepMind's products, and I'll call them products because in some time later we'll find that they are being marketed. Because one is your AlphaFold and the other is your AlphaGenome. So people who have used AlphaFold, what we used to do when we did not have crystallographic structures available to us, we used to do something which we refer to as homology modeling based on a particular template, where you used to generate a protein structure used on a template. AlphaFold has given us the opportunity to just go in and type in the name of the protein, type in the name of the organism, or even submit a sequence, and they will generate the structure for us using AI models. But the structure that is being generated, have you evaluated that structure? Because as we have been listening to the other speakers, that there is a requirement of optimizing the test and the training set. So whatever is available to us, based on that, that particular model is getting generated. And invariably, when you are looking at it from a structural analysis point of view, you will find that there are still inherent errors, which indicates that the training set that is being used are still faulty, or there has to be more refinement to those training sets, which we can look into in detail. So how do we optimize assembly? So when we optimize assembly, we find that the essential problem that we all know of is the traveling salesman problem, where a salesman is supposed to sell his or her product by traveling to various different locations. But the criteria is that that individual cannot visit the same location twice. So that is one of the key optimizing issues. And such similar optimization issues are there when we are trying to assemble genomes. Because not all genomes will have a reference. We know that there is a reference guided assembly and a de novo assembly. So majority of the genomes which are getting sequenced for the first time will have a de novo, will require a de novo assembly. So whenever we require a de novo assembly and there is no reference, we are faced with optimization problems. And through the years, we have seen that there has been the evolution of De Bruijn graphs, etc., algorithms which has contributed towards the refinement of such issues. But still we find that there are specificities missing when we are going in for a genome assembly program. When we are submitting the assembled genomes to the curated databases, we are finding that there are issues still with those genomes. And there are optimization issues which needs to be looked into. So that is one of the key aspects that we need to focus on when we are looking for AI-based genomic data analysis. This is an example with what we did with a Shigella species. You are all aware of Shigella. And we were able to identify and isolate a particular Shigella specimen from wastewater niches, which was resistant to 21 different antibiotics. So it was essentially almost a superbug. And when we delved into its genome, we found that apart from specific known targets or known target classes, we were able to identify through pangenome analysis some very specific transporter classes which were specific to Shigella and specifically multidrug resistant Shigella from around the world. So that indicated that the target space was something which needs to be clearly explored when we are looking for designing drugs as well, because we are now designing drugs based on similarity-based principles. But we have to move away from similarity-based principles to identify more drug spaces. So that is one takeaway that we need to look into. Similarly, one of the key applications of AI in genomic data can be in terms of surveillance, because we are aware that, again, which is related to your antimicrobial resistance field, but when we are looking at surveillance data, do we have some specific model which can predict that after a particular flood in a particular area, what will be the extent of disease-causing microorganisms that will increase after a flood? Do we have a model at this point of time? We do not have that kind of a model. Why do we not have that model? We do not have that model because we have not been able to map our forecast data points and identify them after such environmental events. Very rarely in the entire AMR surveillance network that is available in India, you will find very scarcity of data related to such surveillance. And that is something which AI can actually bring out in the forefront. Models which will enable us to predict what kind of disease burden awaits us after a particular event. So that is one of the aspects that we need to look into. Sir was talking about natural language processing. So we are still at our infancy to understand the language of the genome. And when we are looking at the genomic corpus, the contigs form the sentences and the genes and the other aspects form the words. So we are still trying to identify how we will be able to define the patterns. There has been pattern recognition concepts, there has been pattern identification concepts put forward. But as the genome space increases, the types of genomic data comes in, the type of pangenome data comes in, we will have to expand our definitions of the words. We have to increase the definitions of the sentences and incorporate models which utilizes them. And only in that way we will be able to develop a system which will enable us to create models which will be predictive as well as enable us to come up with good data points. And this is, since we are in the Bengal Chamber of Commerce, I thought that I will share the market space, because that is something which drives any innovation, how sellable it is. And if you look at the statistics, it indicates that it is definitely sellable. There are enough segments which can be explored, which can be invested in, using our industry-academia partnership, which will enable us to create and explore and identify areas where which we can focus on. So that is, I think, the future of AI and AI-based genomics data analysis. All the data points that I showed you are generated by the team of scholars. This is my acknowledgement, and I hope I have been able to finish on time. Thank you.
Thank you. Thank you, Dr. Ganguli. very interesting talk, and I'm sure a couple of questions may come out of this. Now we are going to move on to the Zoom is ready. Mr. Sinha, you are there?
Yes, I'm there, Dr.
Yeah.
Dr. Chakrabarti.
Yeah, how are you?
I'm all right, sir. Thank you so much.
Yeah, so as the session is on AI and legal aspects in research, so here we are having another speaker. I'm just going to read out his introduction. Mr. Sinha is a serial entrepreneur with over 20 years of technology experience, built 5 companies in last 12 years with 3 cash exits. He has keen interest in healthcare and have spent time with WHO and UN solving healthcare equity issues in lower and middle income countries. His latest startup is in the space of biotech and healthcare data. He holds 11 technology patents and has built technology for over 20 years in building the tech right from ATM machines, mobile phone tech to agentic AI. So I met Mr. Sinha in our university, and I found that he's at that time in Bangalore having one startup working with that. But after that, I cannot get Sinha with us in BCCI. Last time also we tried. He was in USA, and this time he was in Europe. He's right now in Europe. But anyway, we thought that this time we will get him in the Zoom, because the topic what he has given is data privacy, security, and regulatory compliances in genomic research, which is very, very important. I'm sure you will all agree with me. Over to Shumit Sinha.
Thank you so much, Dr. Tribhujyoti. Humbled by being called for this conference. Thank you so much. And to all the other panelists, my namaste to all of you from London. And great to be in the esteemed presence of all of you. One distinguishing factor to the rest of the community and to all of you is I'm not a scientist. So I'm a computer scientist, and I've done my computer engineering 20 years ago from NIT Bhopal. And so a lot of things that you guys say, I enable that. But I certainly, some of the things that you said has also gone beyond me, has tossed over my head. So apologies if I don't get some of the genomics and transcriptomic work that you do. But I definitely try to enable researchers like you to do it way more efficiently than how you are currently able to do. So I'll quickly start my presentation. Just a moment. So the topic that I'm covering today is on how data privacy regulatory environment impacts all the data that we access today. I hope my screen is visible to the audience. Yeah. All right. So my topic is that how do we, while we are on the path of finding new cures through multi-omics intelligence, how do we navigate the privacy, regulatory, and compliance environment which surrounds the drug discovery space? And a lot of it has to do with patient data. So quickly, what are the problem statements? Drug discovery is expensive. It has been time consuming for a very long period of time. As some of the esteemed panelists have mentioned, that there's a lot of failure that happens at various stages. I think Dr. Chakrabarti was mentioning that you model something in silico, then you take it to in vitro. In vitro, there's a very small selection that will happen. There's a lot of risk. There's a lot of money that goes into this entire process. And hence, there is a very large timeline as well as costs associated with drug discovery. In a very different scenario, clinical trials take a lot of recruitment time because you can't really identify the right patient early on. As Dr. Ganguli was mentioning, there are so many data models which are available. They may not be most efficient because the training data set utilized to train those data models may not be most efficient, and he's seeing certain errors already. So those challenges are there. And then obviously there are data limitations in how much you can access. So I know that majority of the scientists in the room would have utilized some kind of open source data, either from the 10,000 Genome Project of India or the UK Biobank analysis, or from Mimic data in the US, which is coming out of Mayo Clinics, Vanderbilt, Cleveland Clinic, etc. So you have access to a lot of open source data. You might even be able to go to Nature, download researchers and the research data published in the backend for such research papers and download it and try to utilize it. But a lot of this data is not necessarily 100% complete and utilizable in trying to build out a separate model for yourself. So how do we efficiently solve it? I'm just giving you a blank slate to think. If you had hospitals in India or anywhere in the world, if various hospitals, various diagnostic units, diagnostic labs in the country could pool their data into a common repository. So there was a data assimilator who could have pooled all of this data. And when I say data, this has typically 5 dimensions. You have phenomic data, which is basically what is recorded as, EMR, handwritten notes of the doctors, all the diagnostic reports of the patient. So that's the phenomic record of the patient. If you had access to their genomic, transcriptomic, proteomic data, which are the other 3 dimensions, and then if you had the ability to access their tissue or their biospecimens. The reason is, let's say during the process of the therapy on the patient, you were able to perform a small panel genome sequence. But if you had the tissue available with you or the blood sample available with you or the CSF available with you, you can potentially redo a transcriptome, a whole transcriptome, a whole genome sequence on that tissue. Then you actually unearth more data points which may not be clinically relevant immediately for therapy of the patient, but could actually lead to more scientific development. So if someone had the ability to assimilate all of this data and then make it available to pharma, biotech companies, CROs, research institutions to perform additional analysis on it and come out with novel drugs, that could be an efficient model to solve this problem, right? But this problem, while it looks solvable on a slide when you make it, it runs into various laws that it has to test and also prove to them that you are not infringing on various rights of patients, data sovereignty of a country, data security of a country, national security of a country, and so many other things that come into the picture. So what are these laws that currently impact data? So in India, if you're sitting in India, you have to ensure that you are covering DPDPA, which is Data Privacy and Data Protection Act, which is Digital Personal Data Act, Data Protection Act, which has already been launched in India. It became an act about 1.5 years ago, and earlier in Jan, the notification also came out. So now it is we are all legally bound by that particular act. You have ICMR guidelines. You have Department of Health, or HMSC, how we know it. You might also, if you are collaborating with external agencies outside India, then you need to ensure that you are HIPAA compliant, you are GDPR compliant. In case you are working with organizations in the Middle East, then NDMO kind of offices for Saudi Arabia you need to be compliant to them. If you are collaborating with Taiwan, Philippines, Malaysian kind of countries, then PDPA, which is their Local Data Privacy Act, you need to be compliant to them. So a lot of law you need to comply to, and not only in the country or the organization where you are processing the data, but also in the country and the organization where you are taking the data to get processed, right? So you need to meet laws on all ends. Ganguli wants to publish a paper based on some of this data, he needs to ensure that he has not infringed on any privacy of any patient. He has not had access to any PII or personally identifiable information of any patient. He cannot reach back to that particular patient again So there has to be a fair level of anonymization, de-identification of this data, so that even when he's taking the publication to Nature or any other research publication house, they will check for all of these things before the paper is accepted. For drug research companies, they will do all of this exercise if they need to submit their outcome to FDA for any kind of trial approval. So this becomes extremely important throughout the value chain of data access. So, but all of this, as I mentioned, is easier said than done, right? So what are the core tenets of how do you manage compliance, and how do you ensure that there is a that you meet the regulatory environment of what is set out in these laws? So first of all, at the core of it is patient consent. Have you taken the right consent at various times of how the patient has engaged with you? So have you taken a consent for anonymization and de-identification of their data? Has explicit consent for utilization of their biospecimens been taken? So all of these need to be factored in when you are utilizing any of the real world data out of the 5 parameters that I mentioned to you. You need to manage ethics approval in case the data is being utilized for clinical trials, or it is being utilized for retrospective analysis of data. You might still need to take HMSC ICMR guideline and HMSC approval according to ICMR guidelines. You definitely will need to take ethics committee approval from the hospital or an independent ethics committee for ensuring that patient confidentiality is maintained. Now, here is the interesting aspect. AI can actually not only unearth, biomarkers and help you identify new data which can solve problems for diseases, but AI can also help you extract and structure this data. So imagine in a single hospital, let's say Sister Nivedita University, you have an EMR where the data is stored. You could have notes, handwritten notes by the doctors. You could have a LIMS, a lab management system, which is storing all the diagnostics reports. You could have an NGS lab, which is producing genomic, transcriptomic, proteomic data. You could have IHC slides. You could have H&E slides stored in different systems within a single hospital. An AI system can actually accumulate all of this data through RAG algorithms and assimilate it and create something called longitudinal patient histories. Further to that, we can use AI-powered tools to do anonymization and de-identification at very large scale. So this data, when a longitudinal patient history is created, you can utilize it to ensure that you have done anonymization through the entire longitudinal patient history, along with DICOM images. You can de-identify them. NGS data, you can de-identify them. All the kind of slides, you can de-identify them, and so on and so forth. You can also ensure that biospecimens, whatever excess biospecimen has been kept. So some biospecimen has been utilized and the reports have been produced for the patient. Today, ICMR guidelines say that excess biospecimens can be easily utilized for research as long as they are anonymized. So you need to ensure that biospecimens are anonymized and safely secured in a biobank. And then last of the things is that you need to ensure that your technology environment in which you process all of this data is not using LLM models from outside India, because there are certain restrictions, and rightly so, for national security and data sovereignty, which are already built into the DPDPA, that restricts your ability to send such patient data to LLM models, to OpenAI or Grok, etc., to churn them. So how do you manage that technology environment at an infrastructure level becomes extremely important. So, and then this data, even if it comes to you, right, it is not going to be utilizable very quickly. I think Dr. Ganguli also mentioned that there could be different ways the column headers are available in a single source versus a different space has a different way of storing that data. So how do you ensure that there is a standardization of all of this data? So there is something called OMOP standards, which is done by OSDSI. There are HL7 and FHIR standards. Then there are codes like ICD-10 and SNOMED CT that you can apply to standardize multi-source data into a common repository for utilization by health researchers. So I'll quickly run through this. At Omics Bank, we do this, consistently now across hospitals in India and in Asia. We bring all of their data sources into a single secure processing vault where all kinds of data, including images, PDFs, handwritten notes, scans, JSONs, XMLs, emails, NGS data, or DICOM images, all of the BAM files, FASTQ files, we can create a repository and process all of this data, make it anonymized, de-identified so as to meet compliance norms, and create an anonymized data lake, which is compliant to DPDP, HIPAA, and GDPR laws. And we can then process this once the data lake is created. We can do semantic classification, mapping for correlations. We do quality checks and then standardize it against ICD, SNOMED, HL7, FHIR, and OMOP CDM. And then we also create longitudinal. Now that the data is already de-identified and anonymized, we can now run it on models from external sources to create longitudinal patient history, and metadata can be extracted for quick query and research. And this query engine we already have made available to hospitals and researchers within the hospitals. So you can write complex queries on our system, which allows you to write something like patients over 50. So various demographics, diagnostic information, even biomarker information to create a complex query, and it can give you patient information very quickly. And then you can choose and select patients and drive industry collaboration with biotechs, pharmas, CROs, and research institutions. This is what we are able to today already do with a lot of hospitals in India. We have already implemented in about 12 plus large hospitals in India. We have 10 plus customers. We have filed 11 patents. We have processed about 12 million patients' data. Today, we have access to about 40 petabytes of data from these patients across phenomic, genomic, transcriptomic, proteomic, and biospecimen samples. And we are just 6 people. Out of 6, all 5 of us are AI engineers, and 1 of us is a doctor. So this is what we do. But coming back to the point on data security and the regulatory environment, I think it is extremely important for us to understand that every country will have a different way of operations. There are different sensitivities around patient data, and we need to conform to all of these things, right? And these things today can be conformed through very good technology architecture where you're processing the data. So as long as you are maintaining a very good technology environment and an architecture which meets the local law, when we are saying in India, we need to maintain legal applicability of India. So as long as you are able to maintain a very good technology environment, you do have the compute power as well as the resources to be able to create something like this and make it available for a lot of researchers and people across the country. You might have heard my closing remarks are. You might have heard that some of our government institutions have been recently saying that there is not enough research being done in India, not enough papers being done. I think one of the reasons why some of these things are not happening because you don't have a singular source to access. Now with Omics Bank, you have a singular source to access all of this data and produce research locally. Thank you so much, everyone, for the opportunity, and happy to take any questions.
Thank you, Shubhit. Really, we are very, very happy to have you, at least what you are doing over here, and the other people can get to know about it. But now I think we are almost finished this session with all the sports speaker, and this is open for the questions. So we can take questions from the audience and one after another. Questions from the audience. Looks like people are tired. You don't have any question. Yes.
Yeah, all 4 talks are very illuminating. I have a question to Mr. Sumit. That when you have the samples biobanked, okay, and you got the ethical clearance for say open using, for any kind of research in future. Now, after a few years, you want to use that samples for another one of the research particular application. Do you need to get re-consent from that patient?
So, ICMR guideline, recent ICMR guideline, which came out in February 2024, says that if the biospecimen has been anonymized and excess biospecimen is available, it can be utilized for research without re-consent of the patient. So ICMR guideline is pretty clear in this aspect. And so if multiple research projects are running, as long as the biospecimen is de-identified, anonymized, it can be utilized for multiple projects.
Okay, thank you. My next question is that I don't know your company is a not-for-profit or a for-profit organization. I just wanted to know that data that have been generated from a patient samples or patients various tests, and that data is now you have curated and now can be used for research or maybe for some new development. So, do you think that you need to pay back something to the patients from where the data has come?
So a lot of our data gets utilized, and the hospitals who are the fiduciaries or the data repository holders, it is up to them how do they want to utilize the data, and whether they want to monetize it or not monetize it. Most of the research projects that are currently going with our data is directly with research institutions. So there is no monetary compensation going to the fiduciary holder today. But in future, if let's say any such monetization does happen, then it is up to the fiduciary how do they want to pass on the benefits to the patient.
Thank you.
I have one question for Dr. Chakrabarti. As far as lab work is concerned, and if I want to take use of AI and deep machine learning into looking into the protein-protein interaction and what you categorize as, if I want to if I say validate, so that it falls into native or non-native category. Let's say, because most of the time it's kind of transient interactions if I'm dealing with some kind of intrinsically disordered proteins, and which I believe may end up with, if I do some kind of docking experiment, may end up with non-native form, right? Non-native category. That means, is it that AI is telling me to go back and relook, because it may be that what it says is non-native can be something like really happening, and I think I should move forward with that observation. So is it like I'm using the AI, but is it that it's kind of stopping me and want me to go back and relook into that doubt? Does it give any doubt to my data? Whereas it can be like the true interaction, just because it's nature of the transient nature, or I'm dealing with a highly disordered proteins, so therefore I'm unable to validate through AI. So is it a limitation there?
Yes, it's a wonderful question. So that is exactly why we were actually trying to augment the data sets with partially unfolded protein receptor structure. So because if you want to, for example, IDPs, if the database does not have the complexes with IDPs, the machine which have been trained on the data, you will never be able to recognize it, even though it was an actual interaction. So therefore, it will always tell you it is a wrong or non-native one. But if you have a sufficient amount of so-called transient interaction existing in your database, it will identify the particular characteristics features for those complexes. Now, if you give a query which is a similar complex, it will be able to identify as it. So that is the key, that if we can actually augment it by generative AI, because in this case we are purely reliable on the experimentally solved structure. But if we can generate, for example, the cell paper which we have been talking in the beginning, because we are generating the simulated data itself. But if we have one or two, now we can actually generate multiple, like hundreds of such things. So once we have sufficiently populate such kind of transient interaction in our database, the AI model will be able to tell you if you actually give a real query of transient complex. So the challenge is to populate the existing models, and it's a continuum process, because today's models are obsolete in after a year. It has to be always continuously populated.
Thank you very much.
Thank you. I am Professor Roy. I am a physician by training and profession. I have a very least knowledge about this hard things and soft things also. So I was surprised to see that this session was on the legal aspects of this thing, legal and of course the ethical part. But what I know with my small knowledge, the three things are very important here. One is transparency and accountability of data, which is one of the speakers, learned speakers, told about the black box, but that was not told. Secondly, about cyber security, that is probably another thing which is very important to speak about this. And thirdly, in all science, which is very common, that is the bias. That part was also not very covered. So I would have been happier if these three things could have been covered. Am I correct on that?
Yeah, you know that Dr. Sinha, Mrs. Sinha in his last lecture mentioned about the different rules and regulations
Ethical
of the
aspects,
authorities
ethical
and
aspects.
others. Yeah, yeah. So they are actually, if we go through those particular, they have this particular components in this particular different organizations to be coming up with. Regarding the data security part specifically. The other part, the ethics part and the ethics part is mentioned already by the speakers. We thought that actually it's within this particular lecture we are going to have this, and if we go to the next session, we are going to have that particular part also going to be covered. You are absolutely right with respect to the points, and these are actually going to be during the other sessions also and the panel discussions. Okay.
Thank you.
As we don't have any more time, let us give a big hand to all the different speakers. Can I have one last question? Yeah.
AI or machine learning, anything else. But one question comes to my mind, that why making a design and development of a drug or pharmaceuticals, what kind of human resources we will adapt in terms of potential competency? A biotechnologist, or medicine specialist, or a computer programmer? And who will hold the remote? This is number one. Number two, what is the validation of the AIA computer system, machine learning system? How frequently it is being monitored? Do I make myself clear in my question?
Yes.
Thank you.
If I can, first of all, I should make sure that this is whatever the AI or computational part of the drug designing or drug discovery is just a very, very small part of it. The rest, all the parts are actually experimentally, clinically validated part. All it does is basically, earlier if you have to try out, let's say 1000 molecules and drug, now probably it is shortlisting the numbers with a more confidence value. That's it. Everything, that's why even in my talk I said that there's a I mean, already existing stronger regulatory power is there. I mean, we'd have to go through even before doing the AI or doing any patient data. As a previous question came, I was just telling Professor Chattopadhyay that I mean, we have to do a lot of paperwork before you can get the clinical data, get the ethical clearance. It takes 3 to 4 months to get the ethical clearance. And we are bound by the laws that we will never be able to, share those data in form. But then the output is always in multi-layered validation as required. From a computer to a drug, it takes years. It takes, 50 years sometimes it can take. Even it fails most of the time. And always it has to work in experiment, in preclinical model, then in clinical system, trials, then it goes to a patient or even a clinic. So be rest assured, the computer-derived prediction is not going to come to store. It will be always, those regulations will never be violated. That's my point.
Doctor, may I add something to it?
Yes, please.
So I was just last week in a lab in Hungary, a biotech company in Hungary, and they had a in vitro lab, and within next to the lab they had like a team of 15 computational biology scientists who were sitting. And what they were doing is very amazing. It was fascinating to see, and to answer one of the gentleman's question is what will people look like who are doing drug discovery? So the computational biologists were developing hypothesis. Next door, the hypothesis showed certain, let's say, molecules or protein structures, etc. Let's say 15 or 20 molecules which could impact a certain drug. They immediately took those into an in vitro situation in the lab next door, tested out whether those models are exactly how they are supposed to work, right? And immediately fed that data immediately back into their system to improve their model for the next prediction, right? So AI model can never eliminate the requirement of you to having to go to a petri dish or an in vitro process or an in vivo process to ultimately prove your drug to be right. And then subsequently, there are so many other processes. You have to ensure toxicology. You have to go to phase 1, phase 2, phase 3s before the drug even comes out. So as Dr. Chakrabarti was mentioning, there is a very long drawn, very well established process which comes through before a drug is launched in the market. But yes, the human resource nature is going to change. I think you will see more and more biochemists with computational understanding and biologists with computational understanding coming into the picture, because at some point you will need to interact with compute models to be able to make out a readout inference of what is coming out and whether it is proving in the petri dish in vitro or not, and feeding back that data to improve that model and come back to the in vitro again for an improved efficiency or efficacy of the molecule.
Yes. Thank you very much. So with this, we are ending this particular session.
Yes, thank you, sir. Now we have a pleasant task to perform. I request our chairperson, Professor Dr. Dhrubajyoti Chattopadhyay, to please present memento to the speakers on behalf of the Bengal Chamber. To Dr. Saikat Chakrabarti. To Dr. Dwaipayan Bharadwaj. And to Dr. Sayak Ganguli. I request all the speakers and the chairperson for a group photo, please, before we close the session.